Alpha oxidation of fatty acids. In this lecture, we will study about the alpha oxidation of fatty acids, alpha oxidation of phytonic acid, byproduct of alpha oxidation of fatty acids, and the importance of alpha oxidation. Let us start. What is alpha oxidation? Alpha oxidation occur in those fatty acids that have methyl group at the beta carbon, which blocks the beta oxidation. Alpha oxidation is defined as the oxidation of fatty acid methyl group at beta carbon with the removal of one carbon units adjacent to the alpha carbon from the carboxylic end. The carbon unit is removed in the form of carbon dioxide and there is no production of ATP. Now, where does the alpha oxidation occur? Alpha oxidation occur in the peroxisomes. The alpha oxidation was first observed in seeds and leaf tissues of plants. In mammals, alpha oxidation probably occur in brain tissues. Important features of alpha oxidation. The absence of coenzyme linked substrate is not required in alpha oxidation. The fatty acid is oxidized without coenzyme A intermediate. Alpha oxidation is especially useful in the degradation of branched chain fatty acids or phytonic acid. Phytonic acid is the oxidation product of phytol alcohol. Alpha oxidation of fatty acid. Alpha oxidation involves the oxidation of alpha carbon. This is the fatty acid and this second carbon is the alpha carbon and oxidation of alpha carbon occur. In the first reaction, the monooxygenase hydroxylate this alpha carbon and producing the alpha hydroxy fatty acid. This enzyme monooxygenase require Mg plus 2 and NADPH plus H plus. As a result, the alpha hydroxy fatty acid is produced. This alpha hydroxy fatty acid is converted to alpha keto group and this is followed by oxidative decarboxylation converting the alpha keto group into the carbonyl group and the initial carboxyl group is released as carbon dioxide. The result of alpha oxidation of fatty acid. The alpha hydroxy fatty acid can be further oxidized and decarboxylated to a fatty acid which is one carbon shorter than the original fatty acid. Thus, if an odd chain length compound is used initially, an even chain length fatty acid is produced. And this even chain length fatty acid can further be oxidized by beta oxidation. What is phytonic acid? Phytonic acid is a 20 carbon branched chain fatty acid. It is derived from plant alcohol phytol, which is present as an ester in chlorophyll. Thus, its origin in the body is from dietary source. This phytonic acid is present in animal fats and milk products. Phytonic acid cannot be degraded by normal peroxisomal beta oxidation pathway. The presence of methyl group at third carbon blocks the dehydrogenation of the hydroxyl group by hydroxyacyl CoA dehydrogenase. The solution for this problem is the alpha oxidation pathway. Alpha oxidation of phytonic acid. In the first step, this phytonic acid binds with coenzyme A, and this reaction is catalyzed by the enzyme phytanol CoA synthetase. This enzyme requires ATP and Mg+2. In the second step, and as a result of this, the product form is phytanol CoA. In the second step, the hydroxylation of phytanol CoA at alpha carbon occur, and this reaction is catalyzed by phytanol CoA hydroxylase. This is the hydroxylation reaction occur as a result of this enzyme. As a result, a hydroxyl group is attached, and the product form is alpha hydroxyphytanol CoA. In the third step, the enzyme thioesterase hydrolyzes and liberates the coenzyme A, producing the alpha hydroxyphytonic acid. Alpha hydroxyphytonic acid is produced. This alpha hydroxyphytonic acid is oxidized by alpha hydroxy acid oxidase and FAD to produce the alpha ketophytonic acid. And in the last and the fifth step, this alpha ketophytonic acid is converted to pristanyl and releasing the carbon dioxide by enzyme alpha keto acid decarboxylase. As a result of this oxidation of phytonic acid, pristanyl is formed. The pristanyl is then converted to pristanic acid by aldehyde dehydrogenase and NAD, which is then transported to beta oxidation pathway. Thus, phytonic acid oxidation occurs through alpha oxidation pathway. Refusum disease. In refusum disease, which is an autosomal recessive disorder, the defect is probably in the alpha hydroxylation of phytonic acid. And the clinical characteristic of refusum disease include the peripheral neuropathy, ataxia, retinitis pigmentosa, and abnormalities of skin and bones. Significant improvement has been observed when patients are kept on low phytonic acid diets for prolonged periods. Example, diets that exclude the dairy and ruminant fat. For further reading, I would like to suggest the following books of biochemistry. First one is the Biochemistry by S.N. Gupta, and the second book is the Fundamental of Biochemistry by Jain and Jain. Thank you for watching.